A patient presents in the emergency ward, actively losing fluid at a catastrophic rate, up to 20 liters a day. Left untreated, their blood volume plummets, their eyes sink into their skull, and they enter fatal hypovolemic shock in a matter of hours. Based on that volume of output, you would expect their intestinal tract to be destroyed. But an autopsy or endoscopy reveals a medical paradox. The intestinal epithelium is completely intact. There are no ulcers, there is zero structural damage, there is no inflammation. This scanning electron micrograph shows the exact culprit behind this crisis, Vibrio cholerae, specifically the O1 and O139 serogroups. You can clearly see the organism's distinctive comma-shaped structure and the single polar flagellum it uses for motility. To understand how this single-celled organism kills without inflicting structural damage, we have to trace its path from the intestinal lumen down to the cell membrane. We will identify the precise biochemical markers it leaves behind and map the specific molecular pathway doctors use to reverse the damage. Vibrio cholerae operates through a purely functional, toxin-mediated reprogramming of human cellular machinery. The infection begins via the fecal-oral route, usually through contaminated drinking water. Because gastric acid is highly lethal to the bacteria, a massive initial inoculum is required. The sheer numbers guarantee a fraction will survive the stomach and pass through the pylorus. Those surviving organisms arrive in the alkaline environment of the small intestine, the primary site of action for the disease. To avoid being flushed out by the normal flow of digestion, the bacteria deploy a highly specific anchoring mechanism, the toxin co-regulated pilus, or TCP. TCP physically tethers the bacteria to the intestinal mucosa. The organism firmly adheres to the exterior of the epithelial cells, making zero attempt to invade the tissue itself. This superficial grip gives the bacteria a stable, permanent staging ground in the lumen, positioning them directly over the target cells for continuous toxin delivery. This diagram outlines the bacterium's primary weapon, cholera toxin, an AB5 exotoxin. The process starts when the 5 outer B subunits bind precisely to the GM1 ganglioside receptors on the enterocyte surface. Once the structure is locked in place, the central active A subunit breaks away and penetrates directly into the interior of the human cell. Inside, the A subunit targets a specific cellular switch, the GS protein. It triggers ADP ribosylation, a chemical modification that forces the GS protein into a permanently locked-on state. That defective GS protein now continuously activates an enzyme called adenylate cyclase, massively spiking the cell's internal concentration of cyclic AMP. This internal chemical surge directly forces the CFTR chloride channels on the cell membrane to lock open. A massive volume of negatively charged chloride ions dumps continuously out of the cell and into the intestinal lumen. Simultaneously, the elevated cyclic AMP completely blocks the standard pathways the cell normally uses to absorb positively charged sodium. With chloride flooding out and sodium trapped in the gut, the resulting osmotic pressure physically drags the body's water out through the cell membrane to balance the electrolyte shift. The enterocyte is entirely hijacked. It is trapped in a runaway secretory state, effectively draining the patient's circulatory system straight into their gut. Macroscopically, this translates to the classic clinical presentation of cholera: profuse, painless rice water stools and profound dehydration. As the secretory diarrhea accelerates, the massive loss of potassium triggers severe muscle cramps, while the loss of bicarbonate pushes the patient's blood into dangerous metabolic acidosis. To verify the organism causing this strain, technicians rely on a specific culture medium shown here, TCBS agar. The distinct large yellow colonies definitively prove that the bacteria are fermenting sucrose, which is the gold standard indicator for Vibrio cholerae. For rapid bedside identification, doctors examine the stool directly. This dark field microscopy video highlights the organism's signature darting motility, a frantic shooting star movement powered by that single polar flagellum. Final biochemical confirmation is achieved by proving the bacteria are oxidase positive, which cleanly separates them from other common enterics, alongside a positive string test. Linking these lab markers directly to the secretory diarrhea is paramount. The biochemical conformation justifies the immediate aggressive fluid replacement needed before the patient slides into irreversible shock. Proper fluid rehydration alone drops the mortality rate of cholera from over 50% to less than 1%. This diagram reveals the unbroken cellular pathway that makes survival possible. The cholera toxin completely disables standard sodium absorption and forces the CFTR channels to leak, but the Na+ glucose cotransporter, known as SGLT1, remains totally unaffected by the intracellular CAMP spike. This intact pathway is exactly why this standard WHO-formulated oral rehydration salts packet requires specific ingredients. By examining the label, you see it pairs glucose anhydrous directly with sodium chloride. The SGLT1 transporter activates only if one glucose molecule and one sodium ion bind simultaneously, pulling both inside. Pumping them across the membrane reverses the osmotic gradient. This forces water to follow the molecules inward, immediately stopping the systemic fluid loss. By delivering a perfectly calibrated ratio of salt and sugar, doctors utilize this unbroken biochemical pathway to bypass the bacterial toxin and directly reverse hypovolemic shock. If the patient arrives unconscious or is already exhibiting severe hypotension, oral therapy is insufficient. Doctors immediately escalate to the WHO's Plan C, rapid intravenous fluid replacement. This specific IV bag highlights the preferred clinical fluid for severe cholera, Ringer's lactate. It is chosen over normal saline because the lactate component is critical to counter the severe metabolic acidosis caused by the massive loss of bicarbonate. Antibiotics like doxycycline or azithromycin are administered, but their role is strictly adjunctive. They are used exclusively to reduce diarrhea volume and shorten the duration of bacterial shedding. They never replace fluid therapy. Crucially, doctors must never administer anti-motility drugs like loperamide. Halting the physical output traps the toxin-producing bacteria inside the gut, artificially extending the hijacking of the enterocytes and worsening the systemic poisoning. This photograph shows the administration of an oral cholera vaccine, utilizing killed whole cell formulations for vital short-term protection. When combined with boiling, filtration, and municipal chlorination, it effectively breaks the chain of fecal-oral transmission. Vibrio cholerae executes a highly efficient, non-invasive molecular hijacking, but by mapping its exact cellular geography and identifying the transporters it fails to break, modern medicine can bypass the damage and neutralize a historic killer with simple, targeted chemistry.